Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia, and I am very proud to welcome you to this edition of our 2022-2023 Foundations of Biomedical Data Science Seminar Series. This lecture is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since 2016, the Foundations of Biomedical Data Science Seminar Series has focused on the use of data science methodologies, artificial intelligence, deep learning, and statistical modeling as they pertain to different themes in the biomedical and health sciences. This year's theme is data science and the public health consequences of the COVID-19 pandemic, where we will be enjoying presentations on Fridays at noon Eastern Time from leading thinkers on the issues, promise, opportunities, and hurdles associated with understanding the secondary health effects of COVID-19. These include delayed health checkups, treatments, mental health, drug abuse, increased obesity, and the long lingering health effects associated with the SARS-CoV-19 virus. Indeed, today we are excited to announce the opening of our 2022-2023 application cycle and encourage junior faculty members working in areas such as infectious diseases, epidemiology, clinical medicine, population health, as well as data science, machine learning, AI, statistics, and other relevant domains to consider applying. We believe this will be an exciting and engaging program where characterizing the secondary effects of COVID-19 necessitates unique quantitative approaches, but also helps to develop new multidisciplinary collaborations. Participants in our Biomedical Data Science Innovation Lab program will leverage these seminar presentations as vital material for our culminating in-person grant project development workshop to be held at the Lodge at St. Edward's Park in Seattle, Washington in June of 2023. So today, kicking us off, I'm delighted to welcome our speaker, Dr. Orlando Lopez, from the National Institutes of Dental and Craniofacial Research at the National Institutes of Health. Dr. Lopez received his PhD from the Mayo Clinic and his Bachelor of Science degree from Boston University, both in biomedical engineering. He is an alumni of the prestigious FDA Commissioner's Fellowship and an NIH Postdoctoral Intramural Research Training Award Fellow. Dr. Orlando Lopez, he currently is the director of the Dental Materials and Biomaterials Program at the National Institute of Dental and Craniofacial Research, and he contributes to the programmatic efforts to strengthen NIDCR's biomaterials portfolio and basic science translational research as it's dedicated to the restoration and repair of dental and craniofacial features. He's also manages technology and innovation grants under SBIR and STTR programs. Additionally, he oversees interagency agreements with the National Institutes of Standards and Technology dedicated to establishing performance standards for dental materials. Prior to being at NIDCR, Dr. Lopez served as the lead regulatory reviewer for the FDA, where he led medical device approvals in imaging, cardiovascular, and digital health technologies. Kicking us off today with our very first seminar series of this season, Orlando's lecture is entitled Radical Approaches to COVID-19 Testing, Surveillance, and Predicting Susceptibility to Developing Long COVID, or the PASC program. And so we are super delighted to have Orlando, and thank you so much for joining us and getting us started this year, Orlando. We look forward to your talk. You're muted, Orlando, so you'll have to unmute.
Okay, thank you.
There we go.
All right, great. So thank you so much, Jack and Jean, for the kind invitation to participate and that very kind introduction as well. So thank you. So I'm delighted to be here to kick off this series, this really amazing series that the Innovation Lab has put together to discuss some of the highlights from the rapid acceleration of diagnostics program, especially in the radical aspects of development. We've really been very excited and very busy, very productive as well in the past 2 years, addressing this public health emergency at NIH. And so definitely like to share some of the highlights and of course acknowledge significant contributions from across all the different program officers and the different leadership aspects of these initiatives, and of course the researchers that are actually doing the work in the field. So Thanks again, and let's kick this off here. So today I'll be sharing different aspects and mainly at an overview level around the specific aspects of some of the innovation and the charge of seeking innovation in the COVID-19 testing to address the pandemic and the different gaps in that space. Looking at some of the discovery and data consortium and coordination centers that are part of this ecosystem of emerging technologies, as well as provide some key examples that are most relevant to some of the data considerations that we're speaking about in this particular series, and a newer a newly launched initiative on the Long COVID Computational Challenge, which it's open for entries, and definitely encourage this community to participate in that effort as well. So the Rapid Acceleration for Diagnostics COVID-19 testing program, of course, kicked off early on in the pandemic, charge from Congress to accelerate innovation in this space, and particularly address the shortage and the actual limitations in performance of whatever was existing, because this virus was non-existent before, so there was really no targets to go by. But the commercialization piece is also very important, and so a number of different initiatives came about to address this early on in the pandemic, and also looking ahead in terms of long-term developments. What would be some areas that might be more early stage, more complex in nature, that needed some additional resources and time to get to, proof of concept and viable commercialization pathways. But initially, the products that were most readily accessible and that maybe require just some more of the manufacturing upscale aspects went into the RADx Tech program, which essentially addressed a number of the different bottlenecks in the early stages. Then there were other program areas that were primarily focusing on the underserved populations, as well as the testing capacity in clinical testing facilities. The particular program that I'll be talking to today is the RADical, which is aimed to develop advanced, novel, non-traditional approaches with new applications, not just for COVID-19, but also that have the potential in terms of viable platforms for future disease detection and health monitoring, to address potential future pandemics and other aspects of public health. The data management support came to be represented in the context of data coordination centers that support the different aspects of the different programs here. And I'll highlight some of the most recent accomplishments in those spaces. So just to give you a bird's eye view of some of the significant challenges, and also just the programmatic complexity in the early stages, the RADx Tech program kicked off a solicitation, and submissions were accepted at the early stages of the pandemic in April. Several thousand, I believe, applications came in, and then all those projects were put through a pipeline, kind of like a Shark Tank type activity, and very few actually made it all the way, but the ones that made it, definitely got significant support across the board to be able to realize those goals of getting reliable testing deployed. And when we see that, of course, we still have a lot of challenges in that space, and we're trying to address that in different areas. So from the perspective of getting things to market, it's important to consider the overall biomedical product development cycle and the different entities and considerations that are, of course, make part of this puzzle to get something to market. So initially, just at a conceptual level, starting from the discovery phase, ideation of a project, and then going through preclinical assessments. I mean, that's mainly where NIH, for the most part, has a significant role in terms of bioecosystem, innovation ecosystem, as the largest seed funder for biomedical research in the world. And then the projects ideally will iterate through a number of research development cycles, early stage characterizations to a point where preclinical safety can be demonstrated primarily in order to start some human testing. Then clinical testing, of course, starts, and then other agencies get involved, more involved, some earlier than others, but FDA, of course, is needed in terms of the regulatory oversight, and then reimbursement is, of course, very important as well. So initially, clinical factors for product development are really, I think, the key drivers for R&D, especially at the early stages, and things that are looked to go towards that a user adoption, market entry perspective. And just like to highlight the safety piece, because a lot of times innovators want to jump into the effectiveness right away without demonstrating that the prototype is even safe. So it's important to address those aspects. And of course, there's many other layers to consider that are non-clinical in nature, and especially around the business models and ways to market that are, of course, that can make or break projects. This is significant areas of product development that we have been discussing and addressing and trying to support a lot of these technologies so that they can find some streamlined path to market, especially because in the context of the RADx-Radical program, these are very novel technologies that perhaps have never been in market before, or at least not for the particular indications that we're looking to support. So very closely, we've been working with FDA and other agencies to be able to leverage as much data as possible from the ecosystem, but also to come up with optimal protocols and policies that help us support these emerging technologies and accelerate their not just the development, but guided development, so that they will be aligned with regulatory considerations and requirements in the future. So typically, the valley of death for a lot of these technologies, especially in the space of biomedical medical device space, it's the lacking the design and doing the preclinical characterization, verification, validation that can then support representative aspects of the clinical performance. So that's a lot of the considerations that we look for, and the way that we oversee these type of projects, as they accomplish and move forward in their milestones to reach the point where they can either become advanced technologies for clinical testing or maybe actually enter the market. So again, just to highlight, the RADx program aims to support the non-traditional approaches, including rapid detection devices for home-based testing, point of care, that address the gaps. Then that was 2 years ago, but there's still significant aspects of some of the gaps there in this space. But also, of course, we're looking for the future and how some of these platform technologies can be leveraged for future applications. So the portfolio of the RADx-Rad program, is very diverse, both from a technology perspective. So about 50 projects were funded initially in the calendar year 2020, including multiple novel technologies across different detection schemes, different indications, different environments of use, different biomarker targets, and of course, addressing different populations in many different respects. We funded, I believe, 11 or 12 different areas, including the Data Coordination Center. And on each area, it's a portfolio within itself. The one that I oversee particularly is the novel biosensing technologies for the oral cavity, and I also participate as a program scientist in other aspects of the SENT program, which is for detection of volatile organic compounds, as well as some of the predictive prognostic work that I'll be talking to briefly. But so about $100 or so million were awarded to these projects, and we're working with some of the projects that are more advanced in nature. Some of them have received additional support, supplemental funds to get them to the next phase of the R&D, and even to some of the scalability and of the product development, as well as some of the regulatory streamlining of the evaluations there to get to market. So I will highlight a couple of examples from some of the novel biosensing activities as well as the SENT program for scanning for COVID-19 by electronic nose, and then some of the wastewater detection, as well as some of the data coordination aspects as well. So for the enabling technologies, the oral biodevices technologies were really the foundation for the RADx-Radical that we put together from NIDCR, the National Institute of Dental and Craniofacial Research. So we have invested in previous years prior to the pandemic on the development of very novel techniques to leverage the oral cavity as a way of access to not just the oral cavity itself, but the rest of the body, where we can look for potential indicators of overall health and disease, and leverage that accessibility for discovery, diagnostics, and therapeutics. And so the oral cavity, from an engineering perspective, is quite a challenge. There's many different forces, a lot of chemical extremes, temperatures, and of course, everybody eats and sleeps, and everybody has a different, their own routine. And so having an indwelling device that will go into the oral cavity and remain there for some time, and remains robust in its performance over time, it's quite a challenge. But if that can be accomplished and in meaningful ways, it could be a very powerful platform, for looking at health and overall health in the future. And so that came as a platform or baseline for the RADx-Rad call, and we combined with work with our colleagues at NCAT to combine these programs, and came up with a number of different activities, and we are supporting 11 different projects in this area. So this was the call that was put out in 2020, and in the other call from the NCATs on the SENT program, basically looks to leverage the ability of canines, to scent different aspects. I mean, there's been some reports and verifications of canines being able to detect cancer or diagnose diabetes and many other aspects. So we're looking to mimic that capability into an electronic form that could be used for COVID-19, but also, of course, other areas in the future. So looking at very development of these novel technologies, looking at unconventional detection strategies, and access and usability. Some example technologies that have emerged from some of these programs, very exciting around contact tracing with privacy protection. So of course, privacy protection has been the forefront of a lot of data sharing and privacy aspects, for identity and security considerations of users. But at the same time, there's of course significant value in being able to identify and track the potential folks that are infected in this type of scenario. And so this particular development allows for de-identified user inputs, and looking at the locations and being able to actually have user reported positives, where you can see here in the graph there, the color graph, you can see that there's an ability to connect the dots and look at a distance level to be able to assess, whether or not somebody has a level of risk for contracting the disease and should be part of the contact tracing and so on. So this is a blockchain-based platform, and this type of technology has, of course, a lot of different possibilities and potential in this area, but other areas as well. From the SENT program, we have been able to get to a point where some of these technologies can actually detect, COVID positive versus COVID negative patients just from different volatiles that have been analyzed from XL breath. And so this is very exciting. There had been no FDA approved products for volatile organic compound detection or to be used as diagnostics. There was one that came up this past year. And so that kind of sets a precedent for this type of technologies to potentially, move forward and get more advanced. So some of the the different capabilities are basically, through the functionalization of materials that get bound to, specific binding agents or targets related to the COVID-19 virus or some other exhaled breath chemical. And that's put through a process for detection of these volatiles in the vapor form. Some of the protocol for the breath analysis basically covers the taking the breath sample in a bag, the material, the gas materials, then passed through a pre-concentrator of these volatiles. There is some gas chromatography that is conducted. There is different levels of processing, microfluidics, and then typically there is, of course, significant layers of machine learning, AI algorithms that crunch through all these different spectra that are produced for all these different chemical entities, and then they're spit out some type of risk factor or some type of indicator that could help clinical decision on the diagnostic. So this is maybe some of the spectra that come out of this type of technologies, which are then analyzed, and then from there there is a determination of the detection of a particular compounds or the signature of a particular combination of compounds that might be representative of whether or not somebody has the disease or the infection. This is more data looking at non-COVID and COVID. There was even there has been some demonstrations around the detecting differences between the different variants of the virus, and so this is really encouraging. And these researchers have done some outstanding work to get this technology to where it is, and we're very much looking forward to getting this more further developed. Now to shift gears a little bit to the wastewater detection of SARS-CoV-2. This has, of course, been really very innovative. Sorry, there's a alarm going by. So yeah, the wastewater detection aspect, of course, came to be considered a significant tool or potential tool to address the prediction of the COVID-19 infections through more at a public health level, a community level. And so this has been really exciting from a more environmental perspective, and many others as well. So some of the goals for this program have been around the investigation of specific approaches that could aim to inform and optimize not just the sampling, but also the detection of these wastewaters, demonstration and optimization of the sample analysis, and then development of intervention strategies that could actually make best use of this data and the information gathered from it, which in many ways could be very unique, and incorporate that into, of course, more advanced computational techniques that could be used for prediction. So some of the advantages for this approach could be that it could be really broad coverage across different municipalities, local entities. It could be very cost effective, cheaper and faster than clinical screening, so like doing patient by patient. There is some significant demonstrations of indications of wastewater content and correlations to COVID-19, and that has been demonstrated in a number of different reports and papers. And so there's of course significant value here, as well as the ability to complement other approaches and be able to just make for a stronger decision making, the more data that we have from different perspectives, different risk factors. So there were 6 projects that have been funded through this initiative, and they're all moving forward very productively. And so, of course, data is key. So, like I mentioned, there is the RADx-Rad program, has a data coordination center component to it, which supports administration and operations and the logistics of some of the data intake and sharing from across the different projects, and the data management and use, as well as supporting researchers in different ways. And so with that, I'll segue into some of the updates from that the Discovery and the Data Consortium Coordination Center, which is led by Dr. Lucila Ono Machado, as well as Dr. Shu and Dr. Spencer, and they have also kindly provided some slides to provide some updates on this. And so this schematic outlines the scope of work of the RADx DCC. As you can see, the project led by the different awardees, the awarding team is still early in the, well, it's not early, but early in the program. The DCC worked closely with the NIH to arrive at a minimum set of common data elements for CDEs to be collected by all awardees working with human samples. Since they were the DCC, since then, the DCC continues to spearhead the formulation of common data elements for research or scientific domain specific ones, also known as Tier 2 CDEs. Here, the new work process have been developed to support the wide variety of projects included in the RADx-Rad program. The DCC also built tools for the identification and limited linkage of patients for use by the awardees. The identification was focused on safeguarding information related to certain personal identifiable information, which are targeted by the NIH for infectious disease, such as zip code, which reduced the first 3 digits only. The age shifted by plus minus 2 years, and dates of care measurements were adjusted to minus or 5 plus, plus days. It is important to note that the RADx-Rad program early in the definition required all awardees to submit only the identified data according to the requirements approved by the NIH leadership. And this was important as it allowed for the rapid transfer of data to the NIH RADx data hub, and in turn to the researchers who have interest in the data. Then the final, as mentioned earlier, the DCC also provide benchmark samples to awardees so that they can normalize and ensure their programs are verified using a defined benchmark. The DCC, they gathers all the data from the 50 awardees, collates them, verifies that there are no residual personal identifiable informations in the data to a global, and transfers this to a global code book that was developed later in the program, and submit them along with the original submission to the NIH Data Hub, which in turn receives data from other DCCs such as the RADx Tech or RADx App. So here the page provides a quick snapshot of work accomplished and recorded by the RADx-Rad Data Hub. As mentioned earlier, there are 50 awardees, the 49 scientific awardees, and the one is the DCC as part of the RADx-Rad program. Of the 49 awardees, 40 awardees have shared data with the DCC, including about 21 with multiple submissions. In order to facilitate common data elements, the DCC has held over 100 Tier 2 CDE meetings and has made significant progress in some areas such as wastewater, exosome, and scent. The DCC has also shipped close to 500 samples to the awardees for their use to benchmark their work. Another important role played by the RADx-Rad is to provide guidance to awardees in their interactions with the FDA, and the DCC has arranged 22 such meetings and helped the awardees prepare and make best use of the audience with the FDA. The DCC has also helped 34 awardees prepare applications for resources, 10 licensing agreements, and are in place between awardees and commercialization organizations. You can see a few more statistical information in the other slides. So now just a quick summary of the highlights. We have spoken about the minimum of the Tier 2 CDEs led by the RADx-Rad DCC, and they have also provided prescriptive templates for the data dictionary that can be leverage existing or new our new surveys. We have also spoken about the privacy technology and tools provided by DCC. Another important support by DCC has been to help the awardees navigate their institutional IRBs, particularly for organizations that are not academic institutions that have fewer established work processes. Data analysis support and methodologies is provided to awardees, particularly leveraging cross-learning is facilitated by the RADx-Rad DCC. Of course, one of the key roles is to allow the smooth and flawless upload by the awardees and on the NIH Data Hub. The RADx-Rad DCC has been instrumental in working with the NIH RADx-Rad Hub to evaluate new tools, methods, and procedures. The DCC has filled a vital gap to provide tools to the awardees. They need to consistently and correctly meet the identification of their data. And this is used by about 10 or so of the awardees who have patient information of a total of 20 who have patients. The others work with synthetic or non-identifiable patients. The quality assurance tools allow for automatic screening of awardee compliance with metadata, data dictionary, and data files constraints for the RADx-Rad Data Hub. So from a work process improvement, the main work process was to develop a method for building data dictionaries that would function across a wide range of radically different programs. The DCC has made significant progress in this and developed it. The second area is related to the dbGaP registration, as dbGaP is an NIH-established identifier to streamline access to secure data, such as that gathered under the NIH RADx program. The DCC has worked with awardees and has obtained registrations of 47 awardees. And finally, the DCC has developed new work processes that has facilitated flawless upload of the data to the STP, FTP, and is actively engaged with supporting awardees to support and submit their data to other repositories across NIH and others as well. So here are just some statistics, and in the interest of time, I'm not going to go through them, but I kind of mentioned some of those already. And the DCC has definitely played a significant and crucial role in supporting a lot of these projects. Now we'll shift gears to, sorry, this is another from another meeting. I borrowed these slides, but this is for the long COVID computational challenge. And this is another very exciting opportunity and a newly launched challenge prize competition and we will provide a brief overview on the objectives of this challenge and information on how you can participate and the breakdown of the prize purse totaling up to $500,000. Everyone is welcome to participate in this challenge, and we definitely appreciate the members, the audience, spreading the word on this. The submission period was from when it was launched in August 25th, and submissions will be received until December 15th of this year. So I'd like to acknowledge the NIH leadership and all the contributors that have made some efforts to put together and launch this challenge program. There has been a number of contributors from across NIH, many different programs, including I'd like to also highlight and acknowledge my co-chairs for this program. I'm one of the co-chairs for the challenge, as well as Dr. Erin Iturriaga, Yvonne Schulman, and Ju Chen also contributed significantly to this particular program. So the community challenge is an NIH-long COVID computational challenge. It's a community challenge, and a community challenge is a crowdsourced benchmarking effort which aims to accelerate research by seeding and strengthening a research community, and addressing an important scientific or biomedical question. The format of a challenge is broken into three parts. First comes the question, then the build, and then the benchmark. The question is a research question that the challenge organizers pose and is the guidelines that the focus of the research community to the topic that the NIH is interested in. The build is the portion of the challenge where the participants build a method to answer the question. And the benchmark points to the challenge where the challenge organizers judge the build or method that the participants created against a set of gold standards. So the challenge intends to accelerate the innovation of software tools, to improve understanding of risk factors and support the clinical prognosis of long COVID. Now for an overview of the L3C challenge, and just a quick background on the condition. The actual clinical driver for this is that the overall prevalence of post-acute sequelae of SARS-CoV-2, also known as PASC, and as well as long COVID, are currently unknown. But there is growing evidence that more than half of COVID-19 survivors experience at least one symptomatic PASC or long COVID at least 6 months after recovery from acute illness. The long-term effects and persistent symptoms of COVID-19 can affect anyone, including children, and it can develop in people who had asymptomatic mild or severe disease. Reports also reflect an underlying heterogeneity of symptoms, multi-organ involvement, and persistence of PASC or long COVID in some patients. Symptoms of fatigue, cognitive impairment, shortness of breath, and cardiac damage, among others, have been observed in patients who had only mild initial disease. Despite ongoing research and significant need remains in identifying prognostic factors that can reliably support clinical decisions for the management and prevention of PASC long COVID in SARS-CoV-2 infected individuals. The breadth and complexity of data created in today's healthcare encounters require advanced analytics to extract meaning from longitudinal data on symptoms, laboratory results, images, functional tests, genomics, mobile health and wearables, written notes, EHRs, and other relevant data types. Advanced development of software tools and computing capacity has allowed artificial intelligence and machine learning approaches that are increasingly demonstrating the potential to provide insight into patient-level data from large amount of data to better understand the effect of SARS-CoV-2 on patients. To complement other NIH long COVID research initiatives like the Researching COVID to Enhance Recovery program, better known as RECOVER, the RADx-Radical program at the NIH launched the long COVID computational challenge, also referred to as the L3C, to support creative, data-driven solutions that meaningfully advance the current understanding of the risk to developing PASC and long COVID. Challenge participants will utilize the de-identified electronic health record data available through the INCATS N3C data enclave. The enclave is a central harmonized data repository that represents electronic health records from over 74 healthcare centers across the US. And to protect patient privacy, the identified data provides information useful to researchers without revealing any information that could identify individual patients. And further information on the N3C might be available in a couple more slides, but there's definitely the links that folks can refer to for additional information and also to ask questions about some of this program. So in terms of the objectives of the program, the L3C challenge aims to focus on the prognostic problem by developing AI and machine learning models and algorithms that serve as open source tools for using structured medical records to identify which patients infected with SARS-CoV-2 have a high likelihood of developing PASC or long COVID. Understanding risk factors may help clinicians better understand the underlying etiology of the PASC and long COVID condition. Models will be evaluated using patients who have had an ICD-10 code related to the disease and recorded in the data set and labeled as true positive of patients with PASC or long COVID. And future funding opportunities related to this topic may become available if this challenge, beyond this challenge announcement, depending on the state of the science, public health need at that point, and availability of funds for further development of these models and algorithms into more advanced tools that can support clinical decision-making, not just for COVID-19, but maybe for other areas as well. So one of the challenges of the challenge is that we have there's definitely a issue with the definition of PASC and long COVID. The working definition continues to change with new evidence and is challenging given that it encompasses a multi-organ effect and seen in this disease. COVID-19 positives are defined for this challenge as patients who have had a positive COVID-19 PCR test, not just a diagnosis. And we define patients who have had an ICD-10 code recorded in the data set as true positives. There will be further explanation, and there is also a recorded webinar on this. And so I think I'm going to just kind of skip through all this, but most folks are eligible to participate. There is some challenge participation rules that are provided in this website. These are two that we like to highlight. The prize winners are expected to deploy their solution as open source resource to the community, and so that receiving the prize is contingent on that. And there is also requests or requirements for having established data use agreement to be able to use the data within the N3C enclave. So definitely point folks to those resources and just to become familiar with the rules there. So in terms of the N3C repository, the data represents over 15 million patients. It's, I believe, the largest repository in the world for COVID-related data, and that's what we're going to be using. The data set includes information such as demographics, symptoms, and so on, like a lot of the EHR type of information. There's observational medical outcomes in partnership. And the model, the OMAP model, is the one that is used typically for getting the data on and harmonizing it. And the ICD-10 code is basically the ground truth within the database. But folks are able to bring in other creative ways to check against that. That's also very welcome. So there is access to the data, and so establishing access to the N3C enclave, that's the required data set, and people will have access to the sequestered data that has been reserved for this challenge, and so everybody has to go through that process. The timeline, we submitted the paperwork, and so sorry, this is for the N3C enclave. Onboarding of the enclave, and so submissions are due by the 15th of December. The winners are going to be, and I'm going to breeze through this, but there is, of course, judging criteria. The first prize will have a pot of 200,000, second prize 150, the third prize 100,000, and there will be 5 projects that will be selected for honorable mentions of 50K each. And these are some of the resources and links that folks can follow up with to ask, to have more information about the challenge, but as well as to submit their questions. So with that, I thank you for your invitation again and your kind patience. Definitely be happy to address any questions that I can. Thank you so much.
Orlando, thank you so much. That was a really great overview of this kind of really exciting opportunity. Some of the activities which have been going on at the NIH concerning COVID and monitoring technologies and so forth. What an exciting opportunity for anybody who wants to kind of get involved and get access to some of that data from the the N3C in particular. Just as a little aside, Johanna Lumba, who is one of the people that you mentioned on your committee that's putting some of the challenges together. She's going to be taking part in our efforts here through our Biomedical Data Science Innovation Lab. We're hoping to have her come join us in Seattle next summer to talk and about the data and to present it more formally and get people engaged with it. So we're super excited about that. One question I wanted to have for you, or that I have for you, is what are some of the roles for that people who are data scientists that are, what extra things could they do if they were able to get access to this data? One of the things you mentioned was on some of these technologies, some of the safety and the effectiveness of that data, or the technologies which are being produced. Would any of the data be available for people who are so interested to, say, get access to it, available through the DCCs? And like look at elements of safety and effectiveness of these various technologies?
Sure. No, thank you. That's a great question. Well, at this point, I'm not sure exactly if the data is available openly, but it's being acquired. Not all projects are able to contribute in the same way. Some are SBIRs, TTRs, or like small business awards. And some of those projects, of course, have IP protection, confidentiality considerations,
Sure.
and so not all the data will be available. But I think if not available now, in due time there might be some data that might become available from some of the DCC repositories that are being put together. So this is it's been about like a 2.5-year effort so far, and so DCC started from scratch basically, and so they have been working very diligently to structure the intake of data and formulate all the processes and so on. So I would expect that some availability of data might become if not now, down the pipe.
That's fantastic. And the challenge program that you described is super exciting. Will be super exciting for people who are interested in this particular topic. And I'm curious if you anticipate the more challenges happening in the future around other elements that leverage these data.
Oh, absolutely. I think that's something that we are exploring. This L3C challenge is a first challenge in this space, at least for us. And so it's definitely been a learning lesson also for us. I mean, NIH and certain groups within NIH are, of course, very savvy and very experienced running challenges in many different areas, addressing very different topics. So we are definitely learning from this experience, and like I mentioned in the presentation, further future funding might become available around this particular topic. And so it depends on the outcome of this challenge. I guess we'll make some determinations as to maybe having additional calls or leveraging this type of mechanism for other areas. So that's to be determined, but it's a very exciting way to accelerate innovation, especially in a space that is very much in need, but there is very exciting potential technologies that could help address some of those challenges. So it's definitely a great way to leverage and maximize value across the way. So definitely see that as happening more often. I just don't know exactly how that will become part of the process here.
Right. Well, it would just be the timing for your December 15th deadline here for our Biomedical Data Science Innovation Lab activities doesn't quite line up. That would be a little too quick for us. But if there were a second phase that dealt with other dimensions of COVID-related data that's available through the N3C, for example, or maybe something's available from the folks at San Diego from the DCC that were available for a secondary challenge that was due next fall, for example. Our Biomedical Data Science Innovation Lab teams that are going to be forming would be in pole position to be able to compete with clever projects for those challenges.
That's great.
Yeah, I noticed it's a number of you mentioned one in particular project that was looking at kind of contact maps of people who, and using the blockchain technology to kind of anonymously keep track of people and their contacts and whatnot. And that looked like that just from eyeballing the slide, it looked like it depended heavily upon graph theory and network theory. And that is certainly something that's of particular interest to folks in this community. I'm curious if you could say a little bit more about that particular project or any of the success stories that it has had.
Yeah, well, unfortunately, I'm not always overseeing that particular project,
Okay.
but I'll be happy to, to provide the contact for the program officer that is leading that. So I'm not, sort of up to speed on the specifics of that project besides the high level, overview that I provided. But I am very familiar with a lot of the blockchain type technologies, and so in general, of course, they have significant potential in the context of provenance, immutability, and being able to really establish trust in a robust way to have reliable data. So in that context, I think that decentralization of elements, I think sets well for this particular network approach of contact tracing, because that is, of course, an important piece there. The de-identification element, I think that's a unique way also to leverage a lot of this data. And so looking at federated learning systems and graph theory, of course, facilitates a lot of the correlation or the mapping of different data assets or different, information elements. And so I can definitely see, these technologies becoming more and more, part of this platform or this type of platforms for the future as these technologies, of course, become more available, and folks are becoming more in tune with their value, because I think there's still some element of blockchain, in particular, compared to AI, that's still somewhat fuzzy to folks. But nonetheless, I think there's no single like silver bullet. So I think it's the combination of a lot of these technologies that very cleverly integrated can definitely deliver value and address a lot of the significant aspects of what are some of the emerging concerns with this, digital technologies and connected healthcare, including some of the TIPS elements. I'm very involved in one of the IEEE standards development around the integration, interoperability of clinical IoT data and devices. And we were looking to address the TIPS component, so the trust, identity, privacy, protection, security, and safety mode. In addition to that, of course, we need to think about the ethical components and a number of other aspects in that line of development of these technologies, in addition to the safety and the effectiveness, which are sort of the underlying performance requirements. But now that we're bringing in all these other emerging techs, we need to start thinking about other elements on how to build trust, in the ecosystem, and of course by the patients and the clinical users that are going to be relying on them.
Yeah, it was interesting when the contact tracing, the digital technologies. I was on a trip in Europe and contracted COVID when I got home. And I got a notification after I had recovered through my phone that I had been in contact with somebody while I was on my European trip, which was a little too late. So obviously some further technological development needs to happen. But one thing which is particularly interesting is the wastewater technologies. And one could imagine these being deployed in the wastewater management system of a particular building, of a village, a town, and then being scaled up to cities. I'm curious if there are any technologies that will be scalable to be able to assess, not only individual homes, but maybe communities as well.
Yeah, well, that's a great vision, and I believe that that's probably part of the plan to some of this. I think at this point we're still at the high level sort of verification and validation stage. There's been, of course, some very promising reports on this in terms of like being able to use wastewater detection for COVID-19 and being able to predict a new wave of infection, maybe about 2 weeks or so in advance. And so that's definitely very promising. And so from a number of different considerations in the context of public health surveillance, I think that this will just become in time just another tool in our arsenal of of these emerging like novel techniques that are really I think very powerful, and when they are validated, which is I think the significant part of the challenge. Once we have them vetted and well established, I think that this altogether will really change the paradigm of how healthcare is actually practiced in many aspects by accessing reliable information to really enhance decision making. So I think that's a very exciting future ahead of us.
Orlando, I want to thank you so much for summarizing all these different activities that the NIH has going on. These are all very exciting. I think would be very appealing for a lot of our potential participants and as sources of data or sources of inspiration to kind of think deeply about where data science could play a role, some of the computational technologies which could be deployed to help in terms of the technology for early detection, for long-term tracking, for discriminating between different COVID variants, et cetera. It's all super exciting. I want to encourage everybody to consider applying for our Biomedical Data Science Innovation Lab activity. The deadline will be January 8th, and applications are being received up until that point. There'll be a priority for folks who apply early. The part of the program is taking part in a few online activities in addition to these seminar series, as well as being able to take part in our in-person activity to be held in Seattle, Washington next June. Check out our website, innovation.lab.virginia.edu for further information and application materials as well. Once again, Orlando, thank you so much. This has been wonderful, and I hope that anybody who has any questions for Orlando will reach out to him directly. And we want to thank you, and everyone have a great weekend. We'll see y'all soon.
Thank you so much. Have a good